I would like to pass on the stage to Dr. Raghavendran to take the session ahead. Over to you, Dr. Raghavendran.
Thank you very much, Sonalika. Okay, let's move on ahead. And after a wonderful talk on single-cell RNA sequence analysis by Professor Sanjeev Kumar, and I feel a little bit intimidated to talk about the same topic in the context of Tauber BIoinfo Platform and how this analysis can be carried out, and how you can perform such an analysis using the platform's inbuilt tools. Let me quickly share my screen and let's go over the topics. So since Professor Sanjeev Kumar covered most of these in an excellent detail and with lot of information, I'm going to, and also we are a little bit short on time, so I'm going to quickly glance through or breeze through some of the concepts that he has already explained in great detail. So welcome to you all one more time. Let me share, and yeah. So while you had already obtained a lot of introduction and information and explanation regarding single-cell RNA-seq data, single-cell RNA-seq process. So let us go through all of these. This will be a very good opportunity for you also to summarize what you have grasped so far in the last 1 hour or 1.5 hours. So while it might seem that single-cell RNA-seq is just a natural step forward from bulk RNA-seq methods, but the complexity and the heterogeneity of the single-cell data, as you had already gotten some information or gotten some introduction to, as well as the significant reduction in the data quality that can be recorded due to much less RNA material per sample is too high. So as a result, much more emphasis is placed on normalization and advanced statistical methods involving machine learning methods and other dimensionality reduction methods. So let's first understand why we need or how the single-cell transcriptomics is going to be employed. So single-cell transcriptomics represents the profiling of all the messenger RNAs present in one cell or present in a single cell. So the most widely used technologies that is used to perform such profiling are quite different, as you had a lot of introduction to how and different and variety of different ways how these could be performed. So they are widely different than what it is usually done for bulk RNA-seq profiling methods. So unlike the bulk RNA-seq profiling methods, where the sequencing libraries are generated from thousands of cells and an average expression analysis or expression data is what is generated by measuring these bulk RNA-seq, the single-cell RNA-seq technologies isolate single cells by various methods and then generate cell-specific sequencing libraries by marking the RNA content from a cell with a cell-specific molecular barcode. So such a technology enables the measurement of transcriptomic information for a range of thousands or even up to millions of single cells in right conditions, in correct conditions. So let's go over some of the major differences between these two technologies, and that includes obviously the distribution of expression levels measured as an average for bulk RNA-seq, while it is measured at the individual level for the single RNA-seq. And due to this, SC-RNA-seq are more suited to study cell heterogeneity, cell type identification, and the differentiation of cells over time, etc., etc. So single cell technology has been around only since 2009. So in the context of genomics, or in the context of Human Genome Project, which originated in 90s, this is a very recent technology. So relevant publications and protocols that are published demonstrate how this technology has evolved and how this technology still continues to diversify. Because for a fairly recent technology, this has a really a variety of ways how this profiling can be achieved and how this analysis can be performed. So separating tissues into cells as a biotechnological or technological or microbiological protocols has been around for a long time. So for example, it's been used for cell sorting and other techniques that have been around for a long time. But the differences and the advancement comes in how this single cell technology adopts these protocols to understand the state of the cell at expression level in a great detail that has not been explored before. So it allows to study new biological questions in which cell-specific changes in transcriptome are important. For example, cell type identification, heterogeneity of cell responses, stochasticity of or randomness of the gene expression or pattern within the gene expression based on the cell type, and the inference of regulatory networks, gene regulatory networks across the cells, across the time points. So these are very important applications of single-cell RNA-seq methods. And yeah, there are the key steps involving single-cell RNA-seq data analysis is: first, you have to extract the cells from the tissues and isolate the cells one by one. So this is an extensive step, and that can be executed by a variety of different technologies. And Professor Sanjeev Kumar has explained in detail, and we will also go over this in a very brief manner. So some steps follow a bulk seq RNA technology, like library preparation and amplification. These are common for both, but the way in which the single cells are isolating the single cells is performed is completely different from how bulk RNA-seq is handled when the samples are extracted and RNA is profiled. So we have to then perform sequencing using HT high throughput or NGS sequencing methods, and then we have to generate a structured data from these unstructured FASTQ files, and the structured data in this case will be a count matrix, and which will be employed for downstream analysis and various downstream analysis, as you already had a lot of introduction to. So single cell RNA-seq analysis includes several steps of quality control and normalization. Due to the small amount of material per cell and the sensitivity of the sequencing, it is best to identify unreliable data sets, and it is best to exclude this from the analysis of the rest from the downstream analysis, and just use the rest of the data that can be normalized, and that has to be normalized to be able to compare and perform downstream analysis. So broadly speaking, the analysis can be separated into three different parts. So first part deals with the raw reads by checking the quality and aligning it to the reference genome, and then the second part deals with the cell-specific data by quality controlling the cell-specific data and normalizing and eliminating the artifacts, and then by analyzing the resulting data sets for differences and differential expression patterns using clustering techniques and network analysis, which is best suited for time course data. So this is just a very brief summary and overview. So this is going to be a little bit repetitive, so let me brief through it. A big part of single cell RNA sequencing profiling is in the preparation of data. So how can you separate cells for sequencing? That is the bottleneck in which based on which the single cell RNA sequencing technology is being developed. So the methods can be categorized into different ways, but the two most important aspects are the approaches that these methods rely on for capture of the cells and quantification of the cells. So one is the micro well method, which performs or which utilizes a series of micro scale wells that are used to capture the cellular materials. The cells are isolated using pipettes or laser captures, and they are placed separately in these microfluidic wells. So a major advantage of such well-based methods is that they can be combined with fluorescent activated cell sorting techniques, and it is possible to select cells based on the surface markers and based on the differences that one can observe in the surface. And this strategy is very useful for situations where one wants to isolate specific subset of cells for sequencing. So one of the main drawbacks in such a method or such well-based method is that they are often low throughput methods and the material gets wasted a lot by separation, and the amount of work required to get a per cell output may not be considerable. So the another technique based on which this capturing of cells is performed is microfluidic platforms. So they provide a more integrated system for capturing cells and for carrying out reactions that are necessary for library preparation. So these platforms provide a higher throughput than microwell-based platforms, typically around only 10% of the cells that are captured in microfluidic platforms, and thus they are not appropriate if one is dealing with rare cell types and very small amounts of input. So these are the considerations that you have to take or you have to make when you have to choose which method or which platform that you want to employ. So moreover, the chip that is used for microfluidic platforms is relatively expensive, but since the reactions can be carried out in smaller volume and money can be saved on reagents and other chemicals. So the third one will be the idea behind droplet-based methods is to encapsulate, as explained multiple times by Professor Sanjeev Kumar, also in the answers, also in the questions, answers to the questions. So is to encapsulate each and individual cells inside a nanoliter droplet together with a bead. So the bead, as you have been explained, is loaded with enzymes that is required to construct the library. And in particular, each bead contains a unique barcode which is attached to all the reads originating from that particular cell or from the specific cell. Thus, all the droplets can be pooled and sequenced together, and the reads can subsequently be assigned to the cells of origin based on the unique barcodes that is cell dependent or cell specific. So the droplets platforms typically have highest throughput of all the methods that we have discussed so far, and the library preparation costs are on the order of less than $1 per cell. But while the preparation costs are very low, the sequencing costs often will become the limiting factor, and thus a typical experiment typical experiment should involve a lot of coverage, and hence the cost will shoot up in that dimension. So let's look at some of the quantification approaches for single cell RNA techniques. So for quantification approaches, there are two main approaches: the full length and tag based. And the former full length approaches tries to achieve the uniform read coverage of each transcript, because the full length is being used for the quantification. But in contrast, tag-based protocols or tag-based approaches capture only either the 5' end or at the 3' end to which the tags are attached. So the choice of this method is very important and has serious implications in the downstream analysis and what types of analysis that the data can be used for. So because in theory, full-length protocols should provide an even coverage of transcripts, but we will see, or we shall see, or we often see that there are often biases in this coverage. So the main advantages of tag-based protocol in contrast is that they can be combined with UMIs or unique molecular identifiers, which can help us improve the level of quantification that one can achieve from these analysis. On the other hand, using these barcodes or using these tag-based protocols to quantify the genes, we are restricted to one end of the transcript, and this may reduce the mappability, and it may also make it harder to distinguish different isoforms. So so the microfluidics based single cell RNA-seq can be covered in some of the major steps that we are going to discuss in a very brief few slides, 2 or 3 slides. So first, you have to in the preparation stage, the cells are suspended to form complex tissue. Cells are suspended from a complex tissue to isolate these cells into different cell types based on different isolation methods. So the synthesis of primers using barcoded primer beads that attaches to barcodes of these different cell types. So that will actually segregate or specify which cell type or which read that originate from which cell type. And then the microfluidic device forms a major component which captures the cells in oil droplets with which contains barcoded beads, and also which contains the necessary components and enzymes for sequencing protocols or sequencing methods. So next, the barcoding of these molecules inside each cell. Each cell has to be tagged by unique barcode to identify those specific cells during analysis methods, because these barcodes will become the sequences, or will become the tag which will quantify those mRNAs and which will segregate those mRNA to those specific cells. So this has to be performed, and the barcodes are added to the oil droplets together with the cells and buffered, which breaks the cell membranes, and the mRNAs are then attached to the poly T tails of the barcodes, and then these barcodes are then attached to these mRNAs, and then they are proceeded for sequencing. So these barcodes actually contain two parts. Technically, they contain two parts. One of the parts, or one component, is the cell barcode, which is similar to all the molecules in one oil droplet. So in one droplet we have one cell, and all the mRNA molecules from that one cell will have one unique cell barcode. So that identifies the relationship, or that identifies the source of that mRNA molecules from that particular cell. And then we have unique molecular identifier, which will allow each mRNA prior to PCR application to have a separate barcode. So these two components will help in first distinguishing mRNA molecules for different or for specific cell types, and also will help in identifying biases that are introduced during PCR amplification methods. So and then we will have to and then the cells are lysed, and cell lysis is performed, and RNA hybridization is performed. So immediately after the droplet is formed, each cell is lysed within the droplet, and its mRNAs are released. Then the mRNAs are hybridized to the primers on its microparticle surface, and then forms the stamp that Professor talked about, single cell transcriptomes attached to these microparticles. And after the mRNAs are attached, the droplets are broken by adding necessary reagents to destabilize the oil-water interface, and the microparticles are collected and then washed. So the mRNAs are then reverse transcribed into DNAs together in a reaction, and they forming a set of beads called single cell transcriptomes attached to microparticles. These are the stamps in which the transcriptomes are attached to the particles in the center. So, and then we have to amplify these stamps, and the barcoded stamps can then be amplified by PCR methods to necessary level that is required for the high throughput mRNA sequencing methods. So for sequencing and analysis, the resulting molecules are then sequenced using high capacity parallel sequencing, and the reads are first aligned to reference genome to identify the gene of origin of the cDNA, and then the reads are organized by their barcodes. And they're a little bit bigger. For some reason, it's So this is how the pipeline can be initiated in the Bioinfo platform. Yes, a little bit better now. So these molecules, the resulting molecules, are then sequenced in high capacity parallel sequencing, as you had introduction to, and then the reads are first aligned to reference genome to identify the gene of origin of the cDNAs, and then the reads are organized by cell barcodes, and the number of mRNA transcripts that constitute to each gene for each cell is digitally counted. So this is where the UMIs and the cell barcodes come into play, and this will help us avoid double counting of sequence reads from the same mRNA transcripts, and then this will help us build a matrix of digital gene expression measurements that can be established for our, or that can be leveraged for our further analysis. So to build the pipeline of SC-RNA sequences on BioInfo platform, we can select appropriate algorithms that can be chosen based on the sequencing data that we have. The steps involved, the steps include pre-processing and then mapping and assigning reads to transcripts and identify cells using barcodes, demultiplexing, and then proceed forward to quantification, artifact correction, cell-to-cell similarity identification, visualization. So this is comparatively a long pipeline because of the necessary that single-cell RNA technology requires, so that we should eliminate and normalize, eliminate outliers and normalize normalize the expression counts, or normalize the data to be able to enable us to compare different cell types. So yeah, a consolidated pipeline will look something like similar to this, and at the end of this pipeline you can submit it, and then this is going to give us a gene count matrix or count matrix, which can be downloaded from the pipeline outputs. Okay, yeah. So when you open this file, so technically when you open this file, what we have is the columns of the table that we have here are the cells that are represented by the cell barcodes in here, and in the row we have genes or ensemble IDs, and in the intersection we have the expression values of these genes. Each are distinctly of the genes for each distinctly barcoded cell types, and that are given in raw count values. So the goal of the data that is used for this analysis was to identify different classes of cells. This particular data that we are seeing here is used to identify the different classes of cells in the murine amygdala tissue, and thus we will use known molecular marker genes that are necessary to understand which cell types are present in the table that we just generated, or the table that is just generated from the upstream analysis of unstructured data to structured data. So we now restrict ourselves, or we now subset or select only those genes that are known to be markers of different cell types, and we generate a smaller table from this larger table. And then this table has to be technically log normalized and then subjected to principal component analysis. We can examine how the cells, or how distinct cell types are clustered, are separated into different clusters based on the marker gene. So this marker gene, using this segregation or this separation based on the marker gene, will give us an overview or exploratory analysis overview of how these cells separate or cluster in the gene expression count data distribution that we have. So more sophisticated visualization and dimensionality reduction techniques like TSNE and UMAP will be able to visualize and cluster specifically distinct group of cells based on the gene expression count that we have obtained, and that forms the exploratory analysis, and that forms a downstream analysis. After performing this exploratory analysis, we have to move towards downstream analysis, which will be more sophisticated, or which will involve sophisticated algorithms. So once we have the count matrix, and once we have explored our data for the possibility and for the presence of patterns within the data, then we can go and employ a number of computation methods that will help us to perform downstream analysis. And one such method is one such package is SCRNA, and yeah, Professor also talked about SCRNA. So I will tell you how this how this pipeline can be enabled, or how this pipeline, how different components of this pipeline come together in the single cell data analysis that can be employed in Bioinfo platform. So the first few steps is to create or to generate a surrogate objects for each sample, and then to check quality control and select only good quality cells for each sample, and then we have to normalize the data for each sample, and then identify highly variable features for each sample or highly variable genes for each sample, which will specifically define the cell types. So, and then we have to perform some integration, and we have to scale the data so that all the expression counts or all the data from multiple different sequencing cell types are in the similar scale, and then we then perform linear dimensionality reduction methods like PCA, and then we remove not to remove, and we perform this dimensionality reduction PCA to reduce the number of dimensions that we are going to analyze with. And then we go to remove batch effects using harmony, and then we go then we continue to cluster key components or cluster cell types by finding neighbors and by finding clusters and patterns within the expression patterns. And then we also perform nonlinear dimensional detection using UMAP and TSNE, and that will help us and identify cluster in the low dimensional space or cluster in the reduced dimensional space. And these clusters can then be assigned to different cell types based on the marker genes that we have. And then once these clusters are identified, and once these distinct clusters are identified from the UMAP and TSNE and other clustering methods, then we can finally establish contrast for which differentially expressed genes can be identified, and differentially expressed genes can be quantified, and we can analyze and interpret those genes that form significantly upregulated or downregulated for different distinct cell types, and extrapolate that analysis to relevant biological interpretations. So to select only good quality cells, one has to keep some of the considerations in mind, and one basic consideration or one important consideration would be the number of unique genes detected in each cells. And low quality cells or empty droplets will often have very few genes. So filtering it out based on the number of unique genes will become an important filter steps. And while on the other hand, cell doublets or cell multiplets may exhibit or may display an abnormally high gene count. So you have to make sure that you have to filter out both the low quality cells and cells which have more than or droplets which have more than one cells or fused cells in them, so that they will not interfere in our quantification of this single cell RNA analysis. So the percentage of reads that map to the mitochondrial genome will also become very will also have to be removed, and this is done due to the fact that low quality cells or dead cells often exhibit extensive mitochondrial contamination. So hence to be careful and to eliminate those cells, we have to completely remove mitochondrial contamination. For example, in this case, the percentage of mitochondrial contamination in these two cells or in these two samples is almost nil. So we don't have to employ that filter in this case. And the next will become standardization, or next the standardized variance plot of genes. Can you mute yourself? Sanalika, can you help me? Yes, thank you very much.
Yes.
Yeah. So a standardized variance plot will help us identify a subset of genes that are found or that can be identified to be expressed in a very high level from cell to cell in a cell to cell variance. Yes, in cell to cell differences or between different cells, and using this plot we can identify those genes and employ those genes and analyze and interpret and use those genes to differentiate between the cell types and differentiate between how the distinct gene expression patterns differ among those cell types, and how it can be used to characterize, and how it can be used to explain the differences that we observe in the cell type. So identifying these highly variable genes will become extremely important in our downstream analysis. And next comes, after this, we comes one has to perform a non-linear dimensionality reduction techniques or methods like UMAP or TSNE that can be performed based on the top principal components that one calculates, and we can use this to visualize patterns in the data. We can use this to cluster cells that have similar expression patterns, and we can use this to identify differences between these clusters based on their segregation and separation patterns. So from these identification of multiple different clusters to annotation of clusters can be carried out based on the cluster specific marker genes. Yeah, thank you, Sonalika. So annotation of clusters then can be carried out based on the cluster specific top marker genes, which is used to identify cell types in each cluster. So these annotation then, after identifying or after annotating these cluster types, can be transformed or transferred into the clusters that we have identified or we have generated in the UMAP or TSNE non-reduction dimensionality reduction methods, and they can be visualized to give us segregation based on the gene expression distribution. So these expression patterns that govern the distinct cell types can also be visualized as a method or as a dot plot, which represents simply a percentage of expression levels based on multiple samples or multiple cell types, or based on multiple genes that we have, and this gives us the genes that are regulated, upregulated or downregulated between different cell types. So now that we have identified distinct cell types, and we have identified and annotated distinct cell types, and we can go on to perform a differential gene expression analysis between multiple different cell types, and based on these comparisons, one can perform a detailed one can extrapolate that to understand the differences between the cell types, and one can perform a detailed comparative analysis on the cell types or cells from different time points. So it depends on the sample that we have. So so far, to summarize the analysis or the logic of the analysis that one has to employ for single cell RNA analysis techniques, let's take a look at these simplified steps. So we map the reads and we get the BAM or SAM file just like a regular RNA-seq pipeline. And then we map the R codes and we produce a table of molecular counts, which is similar to table of expression. And then we check for artifacts, and then we update the table of expression, and then we normalize the data, and then a similarity matrix it has to be prepared and is calculated based on the molecular counts and based on the cell-to-cell distances based on these molecular counts of these, and that cell-to-cell distances will form the basis in which we can cluster or we can perform trajectory analysis of our data. So yeah, depending upon whether we want to find different groups of cells based on whether we want to find different groups of cells that is present in the sample based on the transcription profiles, or whether we want to analyze the cell development stages or cell development steps over time. So clustering helps us, clustering like this methods help us identify cells that are similar, that have similar expression patterns, and assigns them to a distinct group, as we have already seen. This is often performed using dimensionality reduction technique like PCA or TSNE, and trajectory analysis is the one which is going to help us identify, which is going to help us compare cell differentiation or cells that have different expression level based on the differentiation time, and based on the when or based on the trajectory of these differentiation times. So all these information analysis methods, I'll go back, yeah, all these information analysis methods and the logic behind these methods and the choice of the algorithms that you have to employ and how to do and how to run these algorithms and how to perform the downstream analysis are discussed in much greater detail and in very logic based and modular way in the OmicsLogic platform. And I encourage you all to go through and go through the lessons and check that out and experience the single cell analysis pipeline, so that you can develop an appreciation for the promise and for the potential that single cell RNA-seq analysis has in understanding and in answering some key biological questions that bulk cell RNA analysis cannot be employed for. So I will take questions